we have our cue. Here we go. Perfect. So, Katie, first of all, congratulations.
Thank you.
It's a delight and honor to have you here today at Tel Aviv University, receiving an honorary doctorate. And we are delighted to have you here and spending some time and talking with our students.
Thank you for inviting me.
You're here receiving the honorary doctorate, but can we go back a few years? Can you tell me what you did in your real PhD?
Yes, so 1977 was discovered a small molecule, a 2' 5' link molecule by Ian Kerr in London. And it was an RNA molecule, and it was responsible for the interferon-induced antiviral effect. And so my supervisor thought that if we make this molecule, we will have an antiviral compound. And he said that we need badly, it was 70s, and
Sure.
we know that how important
Of course.
antiviral molecules. And so I had to synthesize it and had to set up testing and all of these
So
things.
this was already in the late 70s. You had begun studying RNA and modifications for the purpose of dealing with viruses.
Yes. And
That's amazing.
I had to set up the assay for, testing antiviral compound. Interestingly, all of my colleagues were organic chemists, and I was the biologist. So, and the rest of my life also that I worked with people who were expert on different fields. So I constantly learned from them. I educated them what I was knowing, and then tried to work together. And I made this compound, and it was antiviral.
What kind of viruses at the time?
I just Symbis, because we were
Symbis.
Symbis.
Symbis virus.
Yeah, and the problem was delivering this molecule to the cell. It was interesting because in this research institute, my supervisor came from the industry, so he was in his mind that we have to develop something which is helpful. And the rest of my life, I always, even very basic things I did, I was thinking, oh, it will be good for something.
Yeah, I totally agree. After your thesis, you moved to the United States with your family.
Yes.
Your husband and your daughter, Susan.
Yes.
Susan, we'll talk about her in a minute. And coming to the States at a period when people are talking about gene therapy, the use of RNA and DNA, what triggered or what actually defined the question that ultimately you solved in doing your modified RNA? What was the question on hand?
So in the 90s, actually 1990, started the Human Genome Project. And everybody was focusing on using DNA to fix the genes which might have a mutation and delivering with virus and other means. And of course, the RNA was not very popular. What happened is because RNA seemed like had only problems. Little protein was made, it degraded very quickly. It was also some immunogenicity. So if you see in the 90s, one paper per year was on RNA somebody used, and never ever they published again because there was problem. It was not enough protein and it was not useful. My thinking was that these people are doing DNA and permanent gene therapy, but most of our disease are aches and pains, and we don't have genetic problem. It is just temporarily we should have, we have a wound, and it would be good if it would heal faster, or the broken bone, and so we can accelerate this if we already knew that what kind of molecule has to be there. And if we deliver a protein, the blood wash away. But if we deliver the RNA locally in the wound, it produces that protein. And that protein is what already we have in our body, but not in the right place, not enough. And that was my idea. So the surprise was when I generated the RNA, and Drew Weissman, my colleague, said that, how inflammatory and immunogenic it is.
The idea is how can we deliver the message for the protein to resolve a local problem.
Exactly.
And then subsequently, Drew Weissman, your colleague at UPenn, brought to your attention that RNA can be very seriously inflammatory and cause a major problem. And then again, you came up with the ultimate solution. So what drove you to think about modified RNA?
So first of all, I was already working 10 years on this mRNA therapeutic problem, that RNA code for a therapeutic protein, and that was for when I work in cardiology and then later in neurosurgery. And I tried to develop a program for stroke therapy. And of course, I felt that this 10 years of wasted time if cannot deliver an mRNA which is inflammatory to a patient. So I was thinking that could it be why it is inflammatory and can I do anything about it? And together with Drew Weissman, we try to understand what is inflammatory. And because we have our RNA inside our cells. So the thinking was that maybe those are also inflammatory if we would take out and put in the human, immune cells.
Exactly.
Maybe they are generic. And when we did this experiment, we found that tRNA, when we put on the human immune cells delivered there, it was no inflammatory response.
So the synthetic messenger RNA elicited inflammation.
Yes.
But the control, the tRNA, was perfect.
Yeah.
So what's the difference between transfer tRNA and the synthetic RNA?
There were a lot because, first of all, the tRNA was short and it had a lot of modification and it had different cap, different ends. So all of them came to mind that what could be. But one suspicious thing was that, 25% of the nucleotides in the tRNA is modified.
I see.
So, and this came to the idea, could it be this modification? But how we could prove it, how we could make it. You cannot order just an enzyme which would modify it, because basically in our body, all of our RNA is made from the 4 basic nucleotides.
Exactly.
And during transcription or after transcription, that is get modification. But you cannot order an enzyme. Those enzymes were not even known in scientific thing that what those are. And so the only thing actually were known to how pseudouridine actually was generated.
Now, pseudouridine, can you explain the difference between uridine and pseudouridine?
Yes. So these are nucleosides, means that there are a base and there is a sugar.
Mm-hmm.
And the base is uracil and the sugar is ribose. And both of them has the same composition, only differently linked to the sugar and the base to each other.
So the chemical linkage between the uracil and the ribose is different.
Exactly.
That's it.
So
Wow.
that's why a long time they couldn't even identify that messenger RNA or in our body also have, because the more weight is the same.
I see. So that doesn't necessarily entail a chemical modification in the composition.
Actually, people calling that chemically modified, not at all. Actually, the enzyme, the pseudouridine synthase, it just flips over.
I see.
And that's it.
Wow.
And anyway,
and that makes all the difference. Drew Weissman and yourself, prior the emergence of COVID-2, were already working on the idea of using modified RNA as a vaccine. What viruses were you looking at then?
So Drew Weissman's interest was in HIV.
Okay.
And actually, that's when we met in 1997. He came from Fauci's lab, and he was telling me that I didn't know who was Fauci, because at that time he was not in the television every day. And he told me also that he was working with these dendritic cells. 1997, I thought dendritic cells some kind of neuronal cell,
of course.
As dendrite. But of course, this is the professional antigen presenter cell. And I learned so much, again, because Drew was an immunologist, so I learned immunology and vaccines and viruses from him.
So you started with HIV?
Yes, he was doing those experiments, and I made the RNA, and that was how we started. And
when we get injected, when we get a jab, obviously you're delivering the lipid nanoparticle encompassed messenger RNA, pseudouracil, or uridine. What cells are actually absorbing and uptaking the new message? Is it the muscle? Is it immune cells. What is your feeling about that?
This is not about feeling,
Okay.
We are in science, so
Okay.
it is a knowledge.
I stand corrected.
So, that's the immune cells. Actually, you don't know. You think that you have in your muscle a lot of muscle. There is a lot of macrophage there.
I see.
Those and dendritic cells. So their job is to pick up things. And this is a particular by nature, 100 nanometer or less. And they are the major cells who will taken up. And then, of course, because the lipid nanoparticle contains one component, the ionizable lipid, which is the adjuvant in the vaccine.
I see.
And so that the cell gets this activation, and they rush to the lymph node. All of these naive T cells sitting there, they are presenting, and that's how it goes.
Now, when you say presenting, the RNA is being translated, and the spike protein is being expressed on the surface of the macrophage?
And the dendritic
and
cells.
The dendritic cell
exactly.
as an antigen-presenting cell.
So they pick up the particle, it gets to the endosome, where the pH goes down, then it escapes the RNA to the cytoplasm, translate to a protein, and of course, due to the activation, the dendritic cells already in action to go towards the lymph node, where they will present to the T cells.
Now, you have your whole career devoted to science and the bench. But I understand that your daughter, Susan, has also devoted many hours to her profession. Can you elaborate?
Yes, my daughter is a rower. I also questioned many times, isn't that boring, just, in the boat and
rowing?
But yes, but she said, Mom, I enjoy it, and the minute I don't enjoy, I will stop doing it. But she was 10 years on the U.S. national team, and when we talked many times, I told her how similar her sport and what I am doing, the science.
Is she as accomplished as you?
She is a rowing legend in the U.S.
Oh,
because,
really?
2 times Olympic champion, 5 times
wow.
world champion,
Wow.
and so I told her, that is like the rowers are rowing backwards and they don't see the finish line, and this is how scientists is. We are just pulling hard, doing our experiments, and we don't even know that. How far the finish line, or there is a finish line. And I told her even we don't know whether we are going to the right direction. So, and also it is like when she got to the women 8, U.S. women 8, she got the 2 gold medal in London and Beijing. And so this team sport, so that 8 girls are there, and there is not a star there, not like in a basketball team that there is a star. Those people had to pull together, and it is not a star in that. Everybody is as important. So it is like when you are working in a company, and everybody has to pull together, and we have to have a product which has function. Somebody will be helped by that, and so that's the goal. And everybody together, no star there. And then we hope that the finish line is close. And then when we are reaching that, we start a new race. That's our job also.
Obviously, you grew up in Hungary, and as a child, do you recall any specific experience or experiment that you all of a sudden realized that this is it, I want to be a scientist?
I mean, as a child, of course, I did a lot of experiment because we were seeding. We had our garden, and we put the seed and see the plant is growing, and we had chickens, and I've seen coming out a little chicken from the eggs, and so many interesting things was around. And of course, my father was a butcher, and I was told that I was watching him to open the animal, and my sister was running away, but I don't remember, but they said that I was watching and seeing what is inside.
Gross anatomy.
Yes, I don't remember, but they told me. And it was when I was 16 years old, actually. My high school biology teacher knew so much. I wanted to be like him.
Mm-hmm.
Any topic. He was just seemed to me at that time that he knows everything.
I see. Your story is obviously one of huge success. It hasn't always been that easy. but can you provide some insights and good advice to the next generation, the younger scientists?
So one of the things is that, I persist because I was not giving up. If your colleagues or people working around you and they get promoted, they get more salary, and you feel that I work more harder and why they, you already took away your attention from something that you can change because you cannot change that. But the experiment is there and do better one and focus on. And that was what helped me. Always focus what I can do. And many things around me I cannot influence. It is not my business. It's happening. Life is not fair. That kind of attitude. Because people said, oh, you struggle. No, I was very happy. I was full control in the bench. I was pouring the gel, purifying from bacteria the plasmid, linearizing, making the RNA, and testing it, seeding the cell, running to pick up the radioactive material, clean a little bit the lab. So I did everything, and I was very happy.
And happiness is really important.
Exactly. You have to enjoy what you are doing. So if you want to be rich, I don't know what you have to do, but maybe science is not for that. But if you enjoy solving problems, just like in a word puzzle, but even you have to take the question, you have to ask the question and answer it. And so you have to enjoy that. So I can suggest every young people that they have to do something that they enjoy. And they try to not to distract themselves, to comparing themselves. And what I told them also, then don't try to do things, doing something for please the boss. No, if your students are listening, not even you, they have to do it because they love it. Because if they do for other purposes, they are set up for disappointment.
And do you have any special advice for women in science?
I don't know whether who is listening today, but if the government listen, I would say that to have women to stay in science and advance, government should have some kind of health care which is affordable, high quality. Childcare is important because then they can go back to, because you cannot go away from this job because then it's so much happening that it is very hard to catch up. They also, have to find a good partner who respect their dream, for the girls. And then, like my husband, was always supporting me and respected that I have dreams and I want to work on it. So that's important, for happy family. And so it is not easy, but if they set up their mind that they want to be scientists, they can have families, they can have career, and so they can accomplish.
I see.
Here I am.
Here you are. And I wanted Katie to say that this has been a pleasure. I've learned much, and I thank you very, very much.
Thank you for inviting me.